Wow.
Okay, good evening, everyone. This is Taxonomy Tuesday from New Jersey Mycological Association. This is where our members share their recent fungal finds. We would normally start with people who want to share from their computer. Does anybody want to begin? No one wants to share from the computer, so I guess we'll go to the emails. Doug, would you like to begin? Doug, are you actively on yet? He's still muted.
Yes, I'm here. Sorry, I just jumped on. I sent in if you call up that Eastern
Yes.
jack-o'-lantern just because it I have a question for other people. I mean, obviously everyone has seen them and they're starting to come out and they're beautiful mushrooms. Yes, it's just to have a discussion about bioluminescence in fungi. And, this is not an unusual mushroom. The stumps of trees and brilliant orange pigment, which is some kind of uh, sesquiterpene that makes that pigment. And also, it has the toxins, they're called illudens, but it has a unique uh, reaction for its bioluminescence, somewhat unique that to fungi, which is a luciferin luciferase. Most of y'all probably know that to get this, you have to excite the luciferin molecule and raise it up to a higher state using some kind of high energy intermediate. And then with oxygen, it falls back down and emits low energy light. And that there are more than 125 species of fungi that exhibit this so far that I could find in a recent paper. Um, I've never seen
Background is
it.
this, excuse me, what kind of background is this picture on? It looks kind of weird to begin with. It looks like on charcoal or something.
No, it's just the way the light was. It's at the base of a tree sitting on some dark leaves. Those are
Okay.
just leaves that fell. The lighting was late in the day at Valley Forge Park. So, yeah, that's all that is. And there should be a photograph of the gills. Yeah, you can see the leaves better here. Yeah, it's unusual, the lighting. I see what you mean. It looks unusual. Are you still there?
Yes, I'm still there. It looks like you have two copies you sent in of the email. Same picture?
I think it's the same picture. I
Yeah,
wasn't sure it sent,
yes.
but
Okay.
I wondered if anyone had seen it, this bioluminescence. I think you have to go out when it's pitch black. I've been at dusk and I've never seen it bioluminesce. Has
I have
anyone?
seen it.
Is it very, very low yellow green? You have to be in real darkness to see it.
Well, I don't think it really had to be in real darkness. I just know our neighbor used to have them on the front lawn, and if I went out at night, 10 o'clock or so, I would walk out the block and I could see this sort of glowish thing going that draws my attention. Um, and it wasn't, we had street lights, so I don't know how often, how close that street light
That,
was.
that's good to hear. I will make an attempt to look at it. Uh, but it's an interesting reaction, and there's a whole bunch of molecular papers on using this the way they use the jellyfish aquarin as a reporter gene to light up cells during development and cell migration. And there's active work in using some of the fungal luciferin systems, which are based on a totally different chemistry using the caffeic acid cycle, totally different than what you see, let's say, in fireflies or in jellyfish. I mean, it's evolved in bacteria, dinoflagellates. I'm trying to think fungi, insects. Um, a lot of different organisms have evolved this mechanism, so it must be important, I guess. The value for the fungus is to attract insects at night, for to spread its spores. It's not really clear what that is. Maybe someone could comment about whether they've seen other bioluminescent fungi and what spectrum colors they emit at night. I know there are a number of them. Has anyone seen any other interesting ones?
Armillaria rhizomorphs, which are mostly buried, so they would have to be dug up, which could happen because of an animal or maybe a heavy rainfall. They bioluminesce sort of bluish green, I think. It's called foxfire,
Yes, I
common
know.
name.
I've heard that. I know there was a famous book in the 70s called the Foxfire book that was sort of arts and crafts from North Carolina, and I guess that's where that came from. Have you seen it, Dave?
Yes, a long time ago, while camping in a remote campsite.
Uh-huh. Is
It actually it was on, I think, roots, like tree roots that got dug up, or maybe they were
uh-huh.
just unearthed by heavy rain. And
Uh-huh.
there was definite glowing at night. Um, Also, I've seen the Omphalotus glow in the dark, but I didn't see it happen outdoors. I took a large clump of very fresh jack o' lantern
Huh.
directly into a closet in a
Huh.
friend's house,
Uh,
and no light at all, no windows, no light at all. I couldn't, couldn't see my hand in front of my face. And it took about 5 minutes or so for my eyes to adjust, or for the maybe for the effect to kick in. I'm not sure, but I've seen it, a few times in less complete dark settings, and it was kind of faint. But the time I took it into the closet, the gills all lit up fluorescent green. It was
yes.
really
Yes.
striking.
Yeah, it's apparently only the gills that has the luciferase reaction. It's not present elsewhere
Oh, okay.
in the fruiting body. That's very interesting. I might try that, Dave. That sounds like a good idea. And I don't have.
Yeah, try to get some. It seemed to work with really fresh, moist material. Um, when they first pop out of the ground, a cluster of them, you'll actually get orange liquid on your fingers if you handle them. That's probably the best time. There's another one as well. Panellus stypticus is said to bioluminesce on the gills. Um, I have not seen any really striking examples of this. One time at one of the out of town weekend forays, somebody collected some, and everybody looked at it outside at night, and there was a very, very faint
Uh,
glow.
it's interesting that it has to cost the fungus quite a bit of energy to make these high energy intermediates to do this. So there must be
Huh.
a strong evolutionary reason for this, which of course is all speculation. By the way, Dave, I saw your paper on with Dave Hewitt on Neolectus.
Oh,
Yeah,
right. He told
in
me he's going to
in
send me a copy of that. Oh,
in, uh,
great. Yeah.
in
Oh, I'm glad it got
Bartonia,
published.
and it got in Bartonia. He's an old friend of mine from the Bot Club, and he's. Have you seen him recently? He's down in D.C. now, working in the FDA.
A few years ago, we met down at one of the colleges in Bethlehem. I think it might have been Moravian.
Uh-huh.
There was an exhibit of the paintings of David Schweinitz, a
Uh-huh.
prominent North American mycologist who did his work in the 1700s, I believe. And beautiful, beautiful illustrations that were hand painted by him. So I
Wow.
went to that, and David was there, and I met him and his wife, and very nice people. He's a really, really, really nice guy. And
No, he's yeah, very nice guy. He was the head of the bot club for a few years, but
ah,
he moved to DC with his wife, and he does work for the FDA device division now.
yeah, yeah. He works for the FDA, right? And I actually met him because of a discussion on Taxonomy Tuesday. Luke Smithson was present when I showed some Neolectea fruit bodies that I found here in not far from where I live. And Luke knew that David specialized in Neolectea because he had taken a course with him at, I think, maybe at Drexel. Um, and so he put me in touch with David, and that got the ball rolling. And yeah, and
Nice.
we have some nice conversations on email once in a while. He's a very knowledgeable guy, um, and he is the authority in North America on genus Neolectea. So
Which I never knew. He never told us that at the botanical club.
yeah, he wrote his PhD thesis on it.
I gathered that from the bibliography on your paper there in Bartonia. But yeah, it's a nice
Yeah.
paper.
Oh,
Interesting.
great. Well, thanks for mentioning. Yeah, he did send me an email. He told me it was coming out, and I think he told me he'll get me like an electronic copy or something to look at. So
Oh, if you ask him, he can probably ask the bot club to send you from the academy the real issue, since you're an author. If
oh,
you, I mean, I would imagine they would do that.
okay, yeah, or I can work something out with them. I can buy a copy or whatever.
Yeah,
Um, whatever. I'd like to have it though.
yeah. It was interesting. He had collaborated with someone in Romania on in that paper also. So he has
Oh, that's right. He did.
yeah,
Yeah.
connections all over, which is interesting. How
I
international.
believe I believe that globally there are only two species in that genus.
Uh-huh.
And now that Australia might make me be wrong here because there's always some weird thing in Australia.
Oh, it says two species.
Yeah, there they are. So Vitulina is the one that I found here in Northeast Pennsylvania in a stand of, I guess it was mostly, geez, at first I thought it was Scots pine. And then I took a tree expert to the spot and he told me he identified like three different kinds of two or three needle pines. And it looked like pitch pine actually was probably the one where these things were associated. And apparently they must be mycorrhizal because they come up every year at the same time, in the same location. Very, the same, like maybe a dozen square meters. That's sort of size area.
Okay, well, thanks for the side discussion, Neolecta. Uh, I guess they're summer
No,
fruiting bodies.
November, December. You don't see them before the end of October. At
Mhm.
least not this Vitulina that I find near Nanticoke, Pennsylvania. The irregularis, I believe, is a little more common and has maybe a bigger window of fruiting. You find it like in the Adirondacks and maybe in Vermont. It's a conifer. These are conifer things. But
Yeah,
I found Neolecta irregularis in the Adirondacks. Oh, it might have been in fall though. It may have been in September or October. I forget.
yeah, Marisol found Vitulina at Franklin Parker Preserve on December 18th.
Yeah. Okay. So it sounds like it probably is a late fruiter.
Oh, thank you, Doug. You have anything else,
Uh,
Doug, to say?
no, it was just to have that discussion about trying to see the reaction, how to do it, and also to talk a little bit about luciferase enzymatic reactions in bioluminescence in fungi.
Yeah, I'll just
Thank you.
add real quickly that a lot of the photos that you see online are taken with a long exposure camera. So, the glow that you see is usually more intense in those photos than I think what you see in real life. But the best way to really um see the bioluminescence is with a tripod and a good camera with a long exposure at night.
Anybody?
Yeah, that should do it. Now, the time I saw it inside the closet, it was really striking. Just to the naked eye, I didn't have a camera with me. I don't think I had really a good enough camera. I don't even think I had a digital camera back then. We're talking 25, 30 years ago.
Yeah, I think you'd have to have the gills very fresh so that the
Yeah.
enzymatic reactions by picking the mushroom don't run down. You'd have to kind of race back home and do that. You wouldn't want to let it sit around half a day, I suspect.
So Sue, click on one of those and look at the photos instead of looking at the observations. Look at the photos for the species. So
I'm
click
just
on
thinking, what's the
that.
probability of one out of 2,000 having the bioluminescence?
Right. So now scroll down and click on the species so that you're looking at the actual species profile. And now, this is for that second photo right there is going to have the bioluminescence.
Oh, beautiful. Wow, it's green. Wow, gorgeous.
And
Is
also
that
mention, since you might be interested in the mechanism, there is a moss that glows called Goblin's Gold. It's not technically bioluminescent. It kind of like uses lenses in the cells to reflect ambient light or something like that. But you might be interested in thinking about that.
are you saying that it bioluminescence or it's some kind of fluorescence or chemiluminescence? You have to have
It
light to
it
activate it?
It grows in like caves or dark crevices. So when there's like just ambient moonlight, any amount of small ambient light, it will it's adapted to reflect that ambient light and basically magnify it, and then it reflects kind of like a greenish glow like this looks like.
That's interesting. Did you say it was a moss?
It's a moss, yeah. It's called Goblin's Gold. We don't have it in New Jersey, but it's in Pennsylvania. It's pretty rare.
Huh. I have to look that up. Thank you. That's interesting.
Thank you, Doug.
Oh, you're welcome. Thank you.
Is Penny on yet? Does anybody want to share from their screen who joined later?
Yeah, I'm here.
Penny, okay.
Yeah.
Well, let me go to the first one that you sent in. Leccinum, Virginia 1.
Yeah, so I thought it was Leccinum scabrum, and Igor changed it to this. I was excited because it's the first time I found something like this, and the third and fourth picture is the tree that it was near, which was a hornbeam, American hornbeam, which is also called musclewood. And I looked it up, and that's a birch, a kind of birch. So that went along with the scabrum. So I don't know what Virginia 01 means.
It means that it's likely an undescribed species, first sequence from the state of Virginia. That's
I
the
see.
notation. So there are earlier collections than that, and we used to call this Leccinum luteum, although it later transpired that Leccinum luteum is a different species. But VA01 and luteum are closely related to each other, albeit their DNA sequences are a few percent apart. I would say maybe, I don't know, 5 or 6. But they're the closest species in the genetic tree if you go to GenBank and blast, one sequence or another. So there's a connection between them, and they're not quite Leccinum. They're probably Leccinellum in the broad sense.
I see. Yeah,
But
because
VA has
the scabers are not very prominent.
Yeah, but that's a variable feature anyway. The color, the size, the density, etc. I'm just saying that this is not a Leccinum in a strict sense. It's more like a Leccinellum. I think it's closer to something like Leccinum or Leccinellum rugosiceps, which is a species by Peck, than it is to Leccinum scabrum or Leccinum vulpinum. Because those last two are the true Leccinum in my understanding in the strict sense. So VA has a yellow cap, whereas luteum, the collection that I sequenced, it has kind of a some shade of yellow brown, but it's not yellow like that. But like I said, they're closely related, but their sequences are very different still. So there's no question that there are two species involved, and this is reproducible. We have a couple of collections of one and a couple of collections of the other, and the two pairs, they are matching almost to a T, whereas VA and O1 and luteum obviously are 6% apart or something like that because of mismatches, gaps, and so forth. Anyway, so that's a species that's found from Virginia at least all the way up to Canada, because Eva Skifik on Mushroom Observer, she collects these quite a lot too. She finds them.
Igor, is this one supported with the sequence?
The Adirondacks.
Yeah. VA is supported with a sequence, so there's more than one sequence, and
but this one here that's identified as
no.
How do you know it's VA01 then?
Because it's yellow.
Yeah, but why it can't be luteum? Luteum's not in this part.
No,
of the
luteum
world.
is not yellow.
Oh, luteum's not yellow.
Well, at least the collection that we sequenced that matched the type were not yellow. They were brown.
Okay. Yeah. Well, that's interesting.
So you are familiar with the species, Dave, because your sequence, your collection got sequenced too. You found VA01 in Pennsylvania.
I found VA. Yeah, it looks familiar. I think I found this thing before, and I probably called it luteum.
Right. But like I said, they are next of kin to each other, but their morphology, I think, is different enough so you can separate them in the field.
Right, I see.
And once Penny mentioned horbeam, I immediately recall that the first collection of VA01, which actually came from our NGMA from over 10 years ago, Luke Smithson found that under hornbeam as well.
Oh, interesting.
But luteum is also growing under hornbeam in Smithville.
Oh.
So
that's funny.
They are birch related, and trees. They're like almost, they're not quite birches, but they're from the birch family. And Leccinum section scabra, as you know, only grows with birch.
Right.
But still, I don't think this is Leccinum section scabra. It's more closely related to what we call Leccinellum in a broad sense. Things like albellum, rugosiceps, and crossopodium, and so forth.
Yeah, it's just interesting to me that luteum is not yellow, and this one is.
Yeah, well, maybe there's some variability, but like I said, thankfully we have sequences of the type, and one bona fide collection other than mine came from Steve Russell, and his is not yellow either. It's the same color as mine. It's kind of warm caramel, kind of brown color.
Yeah, because luteum means, I believe that means yellow, right?
Yes, correct.
Yeah.
You pretty much see yellow on iNaturalist for the luteum.
Well, that's what these people call, because they see yellow, therefore it must be luteum. And
that's what I used to call it.
That's not necessarily the case, because we have collections which are by DNA luteum, which are not yellow.
So they look like it. Okay. So that was yeah. Go ahead.
Yeah. So keep that in mind, that the duality of these two species. Maybe some of these more yellow collections also are luteum. We just haven't figured this out yet. I would like to see more sequences. Before we can conclude that yeah, luteum is not as yellow as VA01.
That's the first observation, and second one. Hemileccinum subglabripes. Well, I can't pronounce the last one.
Subglabripes.
Yeah, Igor also corrected me on this one. I didn't know what it was. I looked at the cap color is brownish. I looked at the gill surface. The underside, which was obviously a bolete, but I thought it looked kind of like a pseuilus. I was just guessing. I looked around and couldn't figure it out. But when it's cut, it's got red in the wormholes. So that, I guess, was useful, and it is darker red at the base. So that went along with Igor's suggestion.
Can we see more pictures, please?
Yeah, I'm trying to get the thing to move.
Actually, they made some really nice improvements to MO in like a day ago. It became much more responsive. So
maybe
yeah,
if you just click on the picture.
Typical subglabripes. And the place is right too. In New Jersey, it's a rare species. We see it once in the blue moon.
Yeah, I see. Oh, you mean because of the red staining on the stipe?
No. I mean, I just know the species. I don't have to look at what it looks like inside. I just know how it looks outside. And
okay,
there, it's a small genus. There's Hemileccinum hortonii, there's subglabripes, there's Enigmaticum, which is undescribed, and then of course there's Ruberpunctum. And I think we only have 4. So, I think there's another subglabripes lookalike that may or may not have a name. So yeah, basically there's probably no more than half a dozen of Hemileccinum species in Eastern North America. So it's a very nice, tidy genus, and all these species are readily distinguishable from each other if you have enough experience, with exception of the lookalike of subglabripes, which either has a code or maybe it's a described species. Like I said, I have to check. Too many boletes to worry about. Yeah, these are fine to eat. Obviously not in a state because it's old and wormy, but they're okay. They're not too bad. Dave probably has more experience with culinary experience with them than I do.
Yeah, when I was a student in Binghamton, New York, I used to find it in the mixed woods in the nature preserve. I don't find it down here in Pennsylvania. Apparently, it has more of a northern distribution and kind of peters out once you get into southern New York. I also find what I believe is subglabripes in Vermont, where it seems to be pretty common.
Yeah, I think it's mostly a birch associate.
That makes sense. Yeah, there was a lot of birch in the area of the nature preserve where I found it. In 1 year, there was lots of it. And then other years, I didn't see it.
In both occasions, I've seen it in New Jersey, one at Stokes State Forest, another one at Meadowwoods. There were gray birches around. I mean, a tree or two. Mm-hmm. So I do believe that these are maybe almost exclusively growing with the birch trees. But yeah, it peters out pretty much in northern New Jersey and northeastern PA, unless you are in the Appalachia up at an elevation, and then you could probably wind up with some collections in Virginia or something like that.
Yeah, but I think there's the thing you mentioned, that looks like it or has been called subglabripes is more
Yeah.
of a southern thing, south of Pennsylvania.
Yeah, there's one from North Carolina. That's the one I sequenced, and then I think Kuo has mistaken that thing for subglabripes because clearly we have different ITS sequences and so forth, so that we know there's a difference between them genetically.
And Chad Hunter agrees that they're good to eat. She's eaten them.
Yeah, they're nice, tidy, pleasant genus. No issues with any of them.
Yeah, that's right. You asked about eating. Yeah, I did eat them. I thought they were pretty good.
Yeah, I would not eat rubra punctum on account of that strange smell. Maybe
ashtray smell.
Yeah, maybe they don't taste so well either. Maybe it goes away once you cook them. I haven't tried them, but I've eaten Hortonii and I've eaten subglabripes.
I've eaten Hortonii. Yeah, it was okay.
Yeah, I think I've eaten them at Colma probably during mycophagy. Not here in New Jersey. No.
And thank you for coming back to us, Penny, after all this time.
And Penny had a third one.
I do have another one, I think.
Yes.
Yeah, I have another one.
You want to send it in or put it in the chat?
I think it's in the same email.
Oh.
Yeah, that one is more questionable on account of kind of compromise for
the first one that says observations. I haven't participated. There's another one in there. The one.
That's a different one. It's only.
So there's three there, Sue. There's two that are tabs, and then there's one that's a link.
Yeah, I don't know how that happened. It just the way it did it.
The
middle.
Try the middle. It's a red port.
Okay, all right. So I got them completely.
No, I was wrong. There's actually two more bullets from Penny I saw in Mushroom Observer. This is just one of them.
Oh yeah. So this one, I've never found this before, and I wasn't sure what it was. I cut the stipe in half, and it wasn't hollow because the color is kind of like Gyroporus castaneus. But I was just looking through the bolete book, and I came upon Austroboletus gracilis and thought it might be that.
I agree.
Yeah,
looks like it.
We'll probably have more than one. One grows with hemlock, the other one grows with hardwoods, unless it's the same species that's just a little bit.
Yeah, this is a hemlock. This is a hemlock dominant area that I was in.
Yeah, and I think there was a paper from some Mexican researchers a few years ago. They worked exclusively on Austroboletus, and they did establish it was polyphyletic. And it seems like the gracilis sequences, or whoever called them gracilis, were claiming in that new genus that's called Hemi-Austroboletus. So I bet you $20 that's where these things are going to be at. This is not a true Austroboletus. This is something else. This is going to be another genus of Austroboletus like mushrooms. So be ready for another transfer from Austroboletus to Hemi-Austroboletus.
You know what, Igor? I think maybe there are two species going by this name, because I think I didn't look at right on the bolete filter, but I did a search on bolete filter at the last Mushroom Club meeting that we had here, and one of the criteria that we entered into the search feature was bitter taste, and this species came up like first or second. I think it came up as
no way.
I wonder if there's one that's bitter.
Maybe, but we saw a lot of gracilis at Nymph.
Yeah,
and I believe that might be But the thing is, I found myself gracilis, which I thought was gracilis because it looked like gracilis. It was growing exclusively in a hardwood section of the woods. There were no conifers around. maybe one white pine, but that was kind of farther out. But I didn't see any hemlock. So, and that's, kind of added more fuel to my theory that I think we have more than one.
Yeah, maybe it's just a mistake on Bolete filter.
Yeah, I don't think that
maybe
these should,
maybe the search engine makes a mistake.
Yeah, I don't think these should taste bitter.
No, they don't, because a long time ago I ate them just to see what they were like, and they're edible. They're not great, but they're edible.
The other one from
Yeah.
Penny is Leccinum rugosiceps.
Rugosiceps, yeah. So Penny, your luteum, what you call luteum, is related more closely to these than to the true Leccinums.
Yeah, this was in Pennsylvania, right in my neighborhood, and I had no idea what they were.
Yeah,
I didn't.
I would call them
realize.
Leccinellum. I would call them Leccinellum
Okay.
because they look like Leccinellum, but now we have that paper that merged everything back together into Leccinum. So for the time being, it's still Leccinum. Rugosiceps. And it's a fine edible. I've eaten these before.
Really?
Yeah, they change color. They turn red or they turn sort of violet.
Yeah, I have pictures
something.
of that. It turned like a light. The third and the fourth picture. The third one is the dark pink color, and then sort of a blackish blue color.
Yeah.
If you wait a while.
Right. They stain pink or reddish first, and then they turn black. Yeah.
I can't get it to change pictures.
Okay. Why can't you
What
just
if you
click
on the picture?
tab?
I am clicking on it.
Click on the picture.
I've done that.
I don't know,
Oh.
Sue. Your
It is
computer
not
should,
responding.
you should just dump your computer and buy yourself a nice.
No, we should dump Mushroom Observer.
No,
don't
Mushroom
have to
Observer
stop.
never
I
gave
would
me
try.
never gave me in trouble.
iNaturalist, but
I
I
think,
find it
Sue,
difficult.
I think it's
There we
because the page that you're on is the image page and
No,
not
I'm
the
not.
observation
No,
page.
no, not true. It's moving now.
Okay, there you go.
I just find iNaturalist difficult, so I haven't really worked with it yet.
Yeah, so do I. Anyway, so these are oak associated. They're not birch or pine or
yeah, there were two
aspen.
oaks. This was in a
These are exclusively oak associated.
Yeah.
Yeah, I'm stuck on the image thing. I can't get it to.
Very funny looking mushroom, I think. Oh yeah, there's this sort of darkish pink. It looked pinker than that picture. You can sort of see it.
Try to eat them. They're great.
Well, I'll tell you something. I did eat, which was against my usual thing. I cooked them up, and my husband got sick that night, and I did cook them a long time. So I don't know if that was it or if it was something else. But I was fine.
You know what?
These
it's
are
not
known.
poisonous. It could be just some kind of a weird idiosyncratic reaction, and it happens with certain fungi. So you never know.
Yeah.
But it was not poisoning. I can guarantee you that.
Yeah, but
because
a
poisonous mushroom will be poisonous to everybody.
These ones in particular, some people don't handle them well, right?
No, I wouldn't say that. No.
Look at the if you can click on the last picture, you'll see the black color that they eventually turn, but it takes a long time.
Nope, it will not move.
All right. Well, thank you for your help, because I never would have figured this one out, I don't think.
Throw your computer into the dustbin.
But actually, Michael Quo, eventually I went on Mushroom Expert, and he has a drawing that looks exactly like this picture right here.
You even put 3 mushrooms in it?
No, I don't mean that. I just mean the way the cap is unexpanded. Like, I kept going back each day and the cap just wouldn't expand. It's very strange.
Interesting.
Okay, there we got it finally. Anybody wants to eat black? Okay. Did I get all of your pictures now, Penny, from the 3 ups?
Yes, you did. Yes, you did.
Okay. All right. Thank you. Does anybody else have anything they want to share? All I have is Dave to go.
I've got one.
Okay, Jason, I'll stop share.
Mhm. This is not my photo. This is a coworker's polypore growing on a stump up in northern New Jersey from about a week ago. These are the only 3 photos I have.
I think this might be something Postia guttulata, or I'm gonna look it up. I'm gonna look up the name. But you see, it's got these potholes all over it. Is this a
Yeah,
conifer stump, by the way? It looks like hardwoods here.
looks like mostly hardwoods.
Yeah, so maybe I'm wrong because I think the thing I'm thinking grows on conifer. Let me find the name.
Jason, was it really hard? Was it kind of squishy?
All I have are these 2 photos. I wasn't there.
Yeah.
What's the one you can squeeze water out of? Do you know that one? It's like Lila showed me. You grab it and squeeze it, and it just drips water.
That's the white cheese stuff, the Tyramyces
Oh, okay.
illness.
Yes. Okay, it couldn't be that.
So Tyramyces thioneus normally has a different, it's more shelved and has a different margin, like a more pointed margin.
And you don't normally see pores. On this one, we can see pores on Jason's pictures.
Yeah.
Postia is not a bad ID.
Yeah. All right, cool. Thanks, Dave.
Hey, Brandon, you said you have a couple to go? That's it for you, Jason?
Yep, that's it for me.
So,
Yeah,
Brandon, you
I can share for you.
okay?
Um, yeah, so this one found on a walk on Saturday. I mean, it's a Hygrocybe. I haven't done much work on it. And it was found during a walk and kind of didn't take the best photos of the thing. Orange cap, yellow stipe. The stipe is hollow. Gills looked a little more purple than this showed. But the idea I put on it, should probably have more purple gills than this, which is purpureofolia, I think it is. Purpureofolia. Yeah, purpureofolia.
Hygrocybe purpureofolia. Yeah, this photo looks like it.
Yeah. I mean, I keyed it out on the Quo. I think Quo has at least what I consider my most useful Hygrocybe key, and it keyed out right to that. So Not sure.
No
And I'm
stain,
not sure.
right? No staining.
Nope, no staining, didn't go black or anything like that. And it was handled, this specimen was passed around to, 10 to 12 people. So definitely no staining. But yeah, that's my best idea there. And glad to hear at least Dave doesn't completely disagree, though I'm sure even that is
It looks like that's probably right. The gills were purple, right? And it's a and you're pretty sure it's a wax cap and not like a Laccaria or something like that.
yeah, the Lacaria thing. I mean, the gills look very Lacaria, but it's got the hollow stipe that you can see
Oh,
here.
okay. Yeah. Yeah,
So
it's that's probably what it is.
cool.
Was this one also found with eastern hemlock, the way Crow expects it?
Nope. Uh, we don't have Eastern hemlock in South Jersey, I don't think, or at least, yeah, I don't think we do. This was found with pitchpine and yeah, pretty much just pitchpine. But I think in that quo description, it says that relationship probably is not well understood, right?
It says it's often, so it doesn't really mean anything else other than that, often.
Yep. Let's talk about Lactifluus or Lacaria. This is one of the real spicy ones. A couple of people tasted it, and that peppery thing just stayed with you and kind of kept growing for a little bit. So um, I don't know these things all that well. You can see it's stained brown. It did have a little bit of um, almost flocose type thing along the margin, kind of fluffy along the margin.
Oh, the
Um,
cottony margin, right? So
yeah,
I think that's subvellereus.
yeah. I mean, there's a couple species that say that's distinctive. So I don't know how distinctive that is.
This one's too old to be distinctive. That aspect erodes.
Okay, yeah, yeah. But real peppery, that one stains
Yeah,
brown, all that.
those are peppery. Yeah,
Yeah,
those are hot peppery. The brown staining I'm not sure of. I'd have to review that. That there's more than one like subvalerius. I think it's subvalerius is what
yeah,
I'm thinking. There's a like regular subvalerius and there's subvalerius subdistans.
yeah. And you can also see the gills are pretty tight here.
Yeah,
Little bit of forking, little bit of short gills.
yeah,
I mean,
forking near the stem usually on these. Yeah, yeah.
yep,
So I think it's subvalerius that has the cottony margin and not um, deceptivus. Did you check?
Deceptivus, right?
Did you check deceptivus? Because it might be
Yeah.
deceptivus that has the
Well,
cottony margin.
I was checking that and Pipratus.
Oh, what's that other one?
Pip Pipparatus, I think it is.
No, piperatus
Right.
has such tight gills that it almost looks like a flat surface.
Okay,
Um,
great.
the gills are really, really close, and I don't think piperatus does it get to be? Oh, it probably could be funnel shaped. I think it has a thinner stipe for the most part, um, and I don't think there'd be brown staining.
All right, cool. Thanks, Dave. So I definitely want to look at this one, which I'm just calling Collybiopsis right now. I think it's probably subnuda or what is it? Bicolor, biforme, one of those two. But
but it doesn't have hair on the stipe. It was found on pitch pine needles, pine debris. Pretty small. Kind of subspace gills. But yeah, Calipiopsis, Gymnopus.
They're cool. Those types have that like little pinkish purple.
Yeah, kind of lilac color.
Is that from hair or is it smooth?
It's smooth. There's almost like a bloom. You know what I mean? It's not hirsute, it's not actual hairs, but there does seem to be a little bit of a bloom or some sort of coating. But these were also very small. I guess you can see my fingers here. None of the caps were more than a centimeter. So I think that rules out a couple of the colybioid, Collybiopsis, Gymnopus type deals. All right, cool. Well, just wanted to share those.
By the way, I caught only a short glimpse of that Lactarius. I don't think any of those species that were mentioned stain that prominently brown on the gills.
Yeah, I think you're right, but also I think you're right on that. I didn't find one that fit that description.
Yeah, the brown staining was a problem for all of the species that I mentioned.
Yeah, and lack of milk, because usually Piperatus and Deceptivus, they are actually Lactifluus, so they bleed a lot, or they milk a lot.
Oh, this thing did. Yeah, all those brown parts, that's from hours before.
Oh, that's from the milk.
I don't know if it's from the milk. I don't know if it's the milk going that color or if it's the gill tissue going that color, but
it's often the latex that stains the gills.
Yeah. But these things bled heavily and easily.
All right, so we can eliminate Russula because that was another.
Oh yeah, it's not Russula brevis.
Yeah, because I know it wasn't that because brevis doesn't stain brown as far as I know, but I just didn't see. I missed most of that presentation. I stepped away, so I only saw the last 5 seconds.
Yeah, the brown stain was a problem. I had no explanation for that.
Yeah. So I don't think this is Deceptivus, or definitely not Piperatus, because the gills are too widely spaced.
There's more than one variety of subvellereus.
But even subvellereus doesn't stay brown like that.
I don't think so. You know what? I have the Melky book right here. I'm gonna.
Yeah, I know what you're talking about. There's a subvellereus variety glossescens, and actually that's a species.
No, that's a Piperatus sub. Right, that's been raised to a species. I believe.
Yeah, I know it's been raised to a species. So all these acrid, hot tasting Lactifluus, they don't stain that kind of dark brown.
Oh, hey, listen! I got the Melky book out. Lactarius subvellereus variety subdistans gills. This is the Bisset book. Blah 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 blah. Becoming yellowish, often with hyaline droplets, staining brownish to pinkish where injured.
Brownish
so
and pinkish.
Far subdistance does stain brown on the gills.
Well, brownish, not that prominently brown.
Well, we're splitting hairs now.
Well, we're not splitting hairs because if you look at a publication that came out many years ago, where you with some trees, Deceptivus is a species group, and there are lots of Deceptivus in there, multiple species.
Yeah, well, I don't think that's deceptive. It's deceptive.
No, I don't think it's. I'm just saying that the whole group is very
Oh.
confused and or confusing, and there are lots of cryptic species in there. We cannot really put the names on these things.
Okay, so Lactarius subvellereus variety subvellereus gills, staining tawny when bruised and drying cinnamon tan. Okay, staining tawny when bruised.
Well, anyway, that whole book is done without DNA, and all these species there are in the sense of beset.
Well, yeah, but we're just trying to give it the best name we can.
Yeah.
Yeah. I mean, occasionally you're going to be able to say, Virginia 01 if there's a sequence. But other than that, if all you have to go on is morphology, staining, gill attachment, et cetera, you're just going to go with an old field guide identification because that's as good as it's going to be.
Yeah, that's why I prefer not to put any specific names on these things unless you're absolutely certain.
You have to, in order to have a discussion, there has to be names. And even if they're outdated, at least you get into a ballpark.
As close as I could get would be AFF. So
yeah, okay, all right, there you go. Put
if we put
Lactarius
SP on all of these, then we'd have thousands of observations that are only to genus, and that doesn't help us. So it's all about getting the best name, and then we can go back and
but you'll be calling one name like 12 species or something, and you'll be wrong about loving them. So what good is that?
It's a group. It's a group. So what? It's just a name being applied to a group, and some of the ones in the group might stain brown, and maybe some others don't. And they're so that technically is, you might be wrong. But
I don't like
have a discussion. We have to be able to put names into the discussions.
No, I disagree with that. It's better not to put a name than to put the wrong name, in my opinion.
Well, I think we can just say it's that or something there, something close to that. And so I think the name has value as long as you qualify, it's close to it.
Attaching the AFF is a good idea.
Yeah.
Because then you're not really making a mistake.
What is AFF?
Has an affinity for it. So it looks like it. It's
similar AFF versus CF. Yeah, there's a big difference.
Right. And what CF?
Could you guys hear me at all when I was trying to talk and share that last photo?
No.
Okay. So I wanted to chime in on Igor. You said, it's better to call it one thing than call it 12 wrong things. But if you call it all one thing and there's 100,
no, I didn't say that. I said that it's bad to call something which it's not. It's better to leave it as a SP, in my opinion.
Yeah, but you don't know that it's not, Igor.
Right.
Well, I know that.
Pretty much. And we'll take the Lactarius conversation. I mean, that thing I did want to mention, the bruising wasn't super dark. It was kind of tawny. So I think, what was the subvellereus, Dave, that you were talking about?
Yeah, the two different subvellereus.
What you're trying to force fit. You're trying to force fit a name on a species that maybe stains tawny. To my eye, it look quite dark brown.
Yeah, but it's better, and it is a good conversation because, in my opinion, it is better to apply that name and then have someone go through those names and say, oh, that one is or that one isn't, than just call it Lactifluus or even worse, Lactarius. And then, you have a couple hundred species just being called Lactarius instead of maybe these 3 or 4 species being called Lactifluus subvellereus. And we can go and work through them that way.
And
I also remember Colin Damnauer's presentation at NEM where he said that over 50% of collections at herbaria have been misidentified. Okay, so you can apply a name,
but is having herbarium specimens that are ID'd to genus
No,
that
no,
that's
ID to
that's not
species,
good either.
and the names are wrong.
Yeah, ID to species. Yeah, there's a lot of bolete. Those are all boletes, and there's a lot of wrong ones, and it's understandable because some of these, especially some of these really old species, the descriptions have evolved. The descriptions have evolved over time, and to the point where we don't really know what some of these things are. But in without a DNA sequence, okay, how many of these things that we discuss at Taxonomy Tuesday have DNA sequence? A very small percentage. What that means is that for the vast majority of things that we talk about, all we're going to be able to say is genus, unless we allow ourselves to apply perhaps a name that refers to a group of similar species. Then at least the discussion is more interesting, and a person can go into a field guide or a species index and check, to see if what was suggested makes sense. Otherwise, all you can say is genus, and that what fun is that,
And if I was doing a research project to try to, get to the bottom of this and describe, Brandon's mushroom as a new species based on those characteristics, I don't want to be searching through iNaturalist for everying that's in Lactarius SP or Lactifluus SP. I can look at Piperatus, Deceptivus, and Subvellereus, and though it further filters the ones that are most relevant to what I'm looking at.
Yeah, that's a good point.
You can compare
like a
as much
lot
as
of
you
the
want, but the bottom line is that it's a difficult group, and you need a lot of sequencing to begin to make sense of the morphology. And just slapping on a name and say this is good enough, well, maybe it's good enough. The more I think about this, the more I believe that we're completely ill-adapted and incompetent about identifying mushrooms, even with DNA, because we have so many species now because of that. And a lot of them look alike. And I'm talking about not necessarily boletes, but things like this and russulas and so forth. And it's really a mess. And we even haven't scratched the surface because all these type collections are still unsequenced, as far as I'm concerned.
Well, this is to me, this is an example, a very extreme example of the open-endedness of physical science. That the door is not closed on what something should be called, and in this case, the door is fairly widely open because of what you're saying, Igor. But still, at any given point in time, there's a best possible way to discuss things in detail and not in gross, virtually useless generality.
Yeah, and applying a name on iNaturalist is different than in an herbarium. But even in an herbarium, when you put a species name on something, even something that's being re-identified, you put a date and a name, so you know who applied that name and what year, and that's just as relevant as the name that they put on that specimen. So you've got to be able to interpret who identifies it and when, and that tells you a lot more than just looking at the name.
And it's right. It's not like right here, right now, we're putting the final name on forever. It's still going to be peer reviewed on iNet and Mushroom Observer, wherever it's uploaded. There's more input to come, right? Probably.
Yeah, because science continues to modify the hypotheses that make up a given subject. And so there's going to be new things that come out about these white Lactifluus mushrooms. And when those new things come out, perhaps we can have a discussion that's grounded in more confidence. But for now, we only have what's available.
Right. And so what I was saying is here at Taxonomy Tuesday, we come up, those who know are trying to put the best name they can on something, and then that's what is put on iNet or Mushroom Observer. But then there's people weigh in on that, on those apps. That's
Yeah.
how they work. So,
And this is one reason why I like Mushroom Observer better, because you can propose at level of genus, and you can put right into your proposal that, you know, this is probably a group that's not well understood, but you can also post a counter proposal and perhaps at lower confidence that accesses that a species name that might very well not actually apply once more is found out. But for the time being, it's a sensible thing to propose.
and especially if you have the specimen and can check that, then I think it's, you know, then someone can say, well, hey, send it to me because I want, you know, I don't think you're right, and let's have it tested, right?
Oh yeah, yeah. Well, then there's motivation then to get some of these things sequenced also. Oh, and by the way, I just want to make sure I mention the subvellereus group in the Bisset book. The margin is described as with fibrils, and the one that has the cottony margin is actually deceptivus. But Brandon's mushroom didn't have like a real cottony margin; it just had some ornamentation on the edge. So I imagine that we can probably say that qualifies as fibrils.
Okay, let's move on from Brandon's shares. Chris, you said you had a couple to share.
Yeah. All right. Right at the beginning of the meeting, I trudged through a bunch of barberry to check this thing out. I saw it driving down the road at 40 miles an hour and had to go back. But it was a good chunk of them. They're a bit old.
Nice.
They have pores or teeth?
Teeth.
Oh, nice.
Maple tree looks like a maple tree. That's usually where I see this.
Yeah. This thing was pretty old. But I see them all the time here. I mean, I don't know if you could tell, but all of this in the background there on the bottom, you can see that's the Delaware River. So, I mean, 300 feet away. And I see them along this stretch. Every time this time of year I'm up here.
Cool. Yeah, I
These are
think
parasites, so are they killing these trees?
I don't know. The one I have one from last year that I am almost 100% sure that I have a picture from almost 10 years ago on the same tree. So if they are killing it, they're doing it real slow.
Maybe they don't kill it. Maybe they can be a slow-moving parasite, right? May not be enough to kill the tree. That might make them successful.
Yeah. Yeah, that could definitely be a thing. And then yesterday, these were on my neighbor's lawn. Or was it before that? No, it was must have been Friday. It was before the foray. Because that's why I was asking if anybody knew the difference between tabescens and the other one.
What does it say? A pestipes or something like that?
Started with a C.
Yeah, it's
Cespitosa.
Cespitosa.
Cespitosa. Yeah, Cespitosa. Yeah.
Well,
I think that's what they're calling
it's now
most of them.
tabescens.
It's back to tabescens?
Well, it's Desarmillaria tabescens, right?
Yeah.
Well, it was Desarmillaria tabescens 10 years ago, but then they changed it to Cespitosa?
Oh, so
Well,
it was it was Armillaria cespitosa, and then it became Desarmillaria tabescens, and now it's Desarmillaria cespitosa.
It gives you two
Um,
choices on Desarmillaria on INAT. I don't even know if that's correct. I mean,
What is
Yeah, their taxonomy is questionable at this point. I feel like they're just throwing names out there without any proof. But I would call this Desarmillaria tabescens personally.
Would you say it's kind of early for these mushrooms, early in the season?
No, we
nah,
saw them at the last foray.
nah,
So,
they have a big time window of fruiting. Sometimes they even come up in late spring, which is it's kind of rare, but it does happen. Were these following a buried maple root?
that lawn has I think there used to be the same kind of maple that I had to cut out of my front lawn on their front lawn. That would have been years ago.
Oh, okay.
Them on this particular lawn before.
Yeah, I usually find them following um, the root system of a big maple tree, and usually a live maple tree, but old and not looking real good.
Yeah.
Often.
Yeah, there's none of that there now, and there hasn't been a different tree in that spot in at least
Yeah.
7 to 10 years.
So there's no trees there now nearby this?
There's maybe 20 feet away, there's a brand new maple that's, maybe, I don't know, like I could
Oh,
almost put my two hands around the trunk and have them touch.
oh, well, that's a pretty good sized tree.
Yeah, I mean it's not a big tree yet, but
Yeah, it's still pretty good size. But these desarmillarias seem to sometimes grow on the roots of a living tree, so it might also be a parasite. I mean, Armillaria malea is considered to be a parasite.
yeah, we have both in my neighborhood in some
Uh-huh.
serious abundance. So I'll probably see these in all different spots around my neighborhood for the next month and a half at least.
you should put up a sign on a telephone pole and say, destroy these. These are parasites.
No, I don't care. That's somebody else's house.
eat
No,
me?
don't go into your trees.
Yeah, once the fungus is on the roots, though,
Yeah,
I'm not sure that
it is
destroying the mushrooms
destroying
is
the.
going to do a whole lot of good.
Right.
Yeah,
It's like the golden oysters. I mean, yeah, you can eat them, but
You can't eat
it's
enough of them, huh?
no.
And then before we were talking about bioluminescence, but there's also fluorescence, which I grabbed this picture from before. This is the edge of a gill of what's the right name on the Hyphaloma? What do they call it? The ones that the common name is brick caps.
Well, there's two of them. At
Ladder-
least two.
Bladeridius.
Yeah.
Bladeridius.
This is the gill edge of one under UV. And it's weird how it's right on the very edge. When they come up this year, I want to do some more with them.
How much magnification are we looking through here?
Oh, this is probably 100.
100. Okay.
Yeah,
Gee,
because
what are those things on the gills, I wonder?
I don't know. It was there. I mean, I'm thinking maybe they're like clusters of like cystidia or something. But like once you get close, the UV kind of messes with the like because there's so much light. It's hard. The closer you get, the fuzzier it gets.
Uh,
It's a weird thing, weird balancing act. But
It's wild.
yeah, I gotta find some more this fall, and then I can play with them some more. Try to figure out how to get that shot up.
Have you eaten those, Chris?
No, I'm not. I'm not terribly confident.
Yeah, you don't want to get that one wrong.
Exactly.
Mm-hmm.
Okay, does anybody else have anything they'd like to share before we go on to Dave's emails?
I could share a couple.
Okay, Hunter.
Okay, I'll do a screen share.
Hunter, do you have that thing that you put on MO that looks like it might be Boletus Roxanae?
Yeah. I think I have it as Veripes. Is that correct? That one? This one? Or no?
This is some sort of Dacromyces or Dacropanax or something that I'm seeing.
Oh.
We don't see your screen yet, Hunter.
Oh, weird. So sorry. Let's try again. Okay, I think it should work. Just
Yes, we're seeing it now.
okay. This one, Dave? Or no?
Yeah, this one. Yeah, it reminds me of Boletus Roxanae, except the pores are really kind of dark. They're kind of brown, and it's hard to see where they're depressed around the stipe, whether they have that kind of stretched out xerocomoid look.
Yeah, I mean, now I believe though, so last week I showed these baby boletes, and
This is not Roxanae. This is an Agilis type. This is a porcini.
So I think it's the Boletus verb.
With the with the pores that color, they look brown.
Sure. Well, look at the morphology.
I will. So, but do you see in that these? As I showed these last week, and we put Boletus veripes on these.
Oh yeah, that's the ones that used to be nobilismus. Now it's variopes.
Right.
That's not the same as this other thing.
I came back to this. It's right in the same area, so that's why I bought the AI given from INAT Veripes name, but that is like the only reason. And I will say, I only have pictures today. I didn't do any microscopy. I didn't do
maybe it is the same thing. I don't know. Maybe they just changed
there's
a lot.
No question about this being a porcini. The question is about species, but I don't see any
Why do you think there's no question? Do you think that the pores are covered?
I think this is probably closer to nobilis, in my opinion. The reticulation is absent, and the pores turn that kind of a yellowish brown color, which is more consistent with that section of the genus.
All right.
Cool. Okay, well, that's exciting that there's other types like right around the Veripes babies that I collected and I ate for the first time. And that worked out. I'm still here, so that's good.
Those were probably really good.
Yeah, they were. Yeah, and I ate the Tylopolis badius too. I mean, yeah, that's it, right? Badius, yeah. Badiceps, that's right. And then yeah, so yeah, I made a little like Russian style bake with sour cream and Parmesan and onions and Parmesan on top, and baked it. It was good. But I didn't eat it all. So because I want to make sure, of course, I just had a little bit. So okay. So nobilis or is the leading guess here, you think? And yeah,
Yeah,
I mean,
I'm not going to disagree with that. That sounds like a good proposal.
okay. I mean,
Yeah,
this
if there's any doubt, you can just put Boletus section Boletus, and that'll be fair enough.
okay. I mean, right. I mean, everything we put on here without DNA testing is all, a guess based on as much as we, can know about the mushroom without the DNA testing. So, that's what I say, you can never know a mushroom with 100% certainty without the DNA testing, but it's good to put a name. So, let's look at these puffballs. This, I think it's craniiformis. This was really weird though. So these were in white pine, thick white pine, and there was some spruce and around as well. And this, is displaying pretty normally. But then this, I don't know if people have seen this before. This brown, is this a hypomyces on here, or there were quite a few that were sort of older and looked like this. I thought it was interesting. I don't think I've seen that before. Has anyone else?
That's pretty strange. Now, those white inside, those ones with the looks like brown leather.
Yeah. I think, I mean, I think they're probably degraded, so they're not bright white anymore, because it seemed like it was a change that occurred. The first one I saw had this big brown cap, and I was like, oh my gosh, I thought it was a big like Amanita brunescens or something. But when I looked more carefully, it was really degraded, but in this with this weird coloration, really different.
So what color is the inside of those brown ones?
Yeah, I mean, so I don't have a cut brown one, so I can't say. I'm sorry. I don't think I cut one, but I guess I could sort of see, because one had a hole in it. It looked similar, just degraded, like not white anymore, but like it had once been white. So dirty white, but I cannot say for sure. So I'll have to go back, it sounds like, and cut a brown one in half. Yeah, so but I'm thinking craniiformis, but I have, like I said, just photos today, and no, I've been able to do zero research. So I also saw this beautiful, and this is now called Macrolepiota americana. And I showed this from the same spot last year, but it was all white last year, so I couldn't really tell that it was the red staining lepiota. But these red droplets were, I mean, the photo doesn't do it justice. They were just like glistening in the sun. And I laid it on the inside of my hat, and it left a stain, a red stain. There's a beautiful ring on it, and annulus, and where's my other pictures? Let's see. Whoops. Oh, yeah, cool pattern. And then here's a close-up of the gills and the ring. And so I know that's what that one is. And I think that's kind of, I think that's it for me tonight. Thank you.
Thank you, Hunter.
Oh,
Thank you.
so that thing with the red scales, that's the thing that used to be, was it Leucoagaricus americanus?
Right.
Wow, that's a very interesting color pattern for
I,
that
yeah,
species.
it's a little bit orange, but you know, it was just, I think, so fresh and wet, and the sun was,
Okay.
yeah, hitting on it in a certain way. And then the camera I haven't quite figured out on my new phone, which I'm actually returning and going back to iPhone. So I think all those things were combined, because now I brought it home. I put it, it got as far as a flower pot on my back porch, and it looks normal now, you know, like you with a darker red, but not a little more drier looking, more normal.
Yeah, was there staining on the gills or the flesh or anything?
I actually didn't even try, but it looked like on the, bless you, on the stipe maybe had a little bit. Well, and what's interesting, I mean, it came out of the last year, it was all white. And I think it was sort of just all a product of the conditions and maybe time of day and freshness. And I'm not sure. But yeah, because I couldn't even recognize it as the red staining last year. But then you guys enlightened me, because don't those have a yellowing when you bruise them at the base or something?
Yeah, on the base of the stipe.
Yeah.
I think so. Yellow or saffron or something.
Yeah, I should check that out. Yeah, because it's right outside, so I can do that.
I think you might want to check a description, but I think
I'll give it a shot. I vaguely remember that, and that was part of how we kind of figured it out last year, I thought. What do you got, Dave?
Excuse me?
Your first one is up on the screen, Dave, the Neoboletus.
Oh, okay. Yeah. Igor, you're still here, right?
Yes, I am.
Yeah, this is so this is where the oak tree is growing with the Boletus subluridellus that grow near it. But I've thought even the last couple of times I found these red-pored boletes in this area, there's also some spruce trees that are close to the oak. In fact, I went there today and just to have a look, and it's really probably only about 5 meters or 6 meters away, the spruce tree from the oak. And yesterday or 2 days ago, 3 days ago, I forget the date will be on the observation, but there were the ones that are I think are pretty clearly Boletus subluridellus. Of course, eventually there'll be Neoboletus subluridellus, but the ones that grow near the spruce, they look different. The stipe looks different, and they're a little bit darker on the caps. And so I made an observation of all the three different kinds. One patch growing near the
I'm not
oak,
seeing any.
another patch growing near the pine, and the ones that hopefully we're going to see here real soon are the ones that were near the spruce. So let's see if we can
she's
get
reminding
those
me
up.
I didn't share my screen.
Yeah, let's see if we can get those up now. These are the ones that were near the spruce, Norway spruce.
Chameleon. That's what this is.
Well, okay, so it's still Boletus subluridellus.
If you want to call it that, sure.
Yeah, well, I think that's right at this point in time. That's the name. Well, you could call it chameleon. I don't know. Has anything been published?
No, I mean, I still prefer to call it the chameleon because A, it's a fitting name, and my name is Neoboletus, whereas the old name is still Boletus, and people get
Okay.
confused now.
All right.
So,
Go through the photos.
until we get Neoboletus subluridellus, I will not stop using my chameleon name.
Okay, that makes sense. But anyway, go through the photos and try. There's some pictures that are a close up of the stipe to show that the ornamentation on the stipe. So there's pictures. There's like 6 pictures. You have to Okay,
Yeah,
not that one. Wait. Oh, that's a different observation. Oh, did I put the wrong thing here? Oh, I got the wrong.
Oh yeah, that's typical chameleon. The bottom line is, if it doesn't look like subvellereus, it got to be the chameleon.
Okay, so you don't think it can be like Brunio squamulosa, or what's that name?
I don't think so. No, too much.
Yeah,
too much shred in this type.
Oh, okay. Because this stipe looks just, and the ones that come up by the spruce are the ones that always look kind of different. And this is one of the main differences is the stipe.
Remember, I think that you're looking at the same organism, and it produces an amazing showing the amazing morphology that the species can attain even within the same population.
Right. Yeah, that is the right picture, though, by the way. I fooled myself there.
You come there every year and you collect these things, and where you have like 1 or 2 collection sequence from that spot, and that's what these things are.
Yeah, well, I've actually sampled several different times. I put a sample into the plates.
Good. Hopefully, we'll come back as a single sequence.
Yeah, and we'll see if it's, maybe we can even see if it's the same population, which it probably is. So we'll have sequences to compare. Hopefully, I mean, this one's been sampled. I've been sampling. I'm not going to sample any more of these, okay? Because I've already got like 6 or 7 samples in the plates, for these guys. So we'll see. Hopefully we'll see. We'll get some results.
I'm confident it's going to come back as a single species.
I'm putting it up here for discussion tonight because I was interested in hearing what you had to say about it. So thanks.
Yeah, thank you.
So I think there was a question a week or 2 ago about how big do chestnut boletes get? So this is not a trick photo. This is a Gyroporus mushroom. It's one of these species in this, Castaneus group, okay, or whatever you want to call it. There's all these different species. We actually have some names now, but you really have to have them sequenced to find out. But look at this one. That answers the question, sort of, how big can they get? That's a big one.
That's wild. Yeah.
Must be a different species.
I think it is. I've sampled it. I did find a big one in a different location a year or 2 ago. I think it was during one of the microblitzes. So I sent it to Steve Russell, and it was a first. It was a first for his database. So it's some, numerically coded Gyroporus. It's somewhere on, it's on MO and on INAT somewhere.
Yeah, as far as I know, Steve's count of Castaneus is at least up to 5.
Yeah, well, this was a new one. He said it was not in his database yet, and it was a big one. Oh, the other one was from Mill Creek Reservoir. That's right, from the which is oak woods. This was in mixed woods. This was growing in moss actually, but probably it's the trees really the reason why this thing was there. I imagine oaks, other hardwoods. It was really quite a mixture in this spot. There's oaks, but there's also other hardwoods, probably birch. I don't know what else is there. There's some that I didn't bother to try to identify the trees that is.
Yeah, I mean, clearly it looks not just size-wise, but the overall morphology looks very different from the quote-unquote Castaneus group.
Yeah, it really does. This is I'm going to guess that this is the same species that I sent to Steve once before, because that one was also really big.
Do you have a sequence for that one?
Yes, there is a sequence for that one, and so he gave it a coded name. It's
And
somewhere. I think it's on MO. I know it's on INET. It should be on both because I immediately import everything to MO.
all right.
Post
Well,
on
I'll
INET.
be interested to take a look at that other observation when you have a chance to find it.
I'll email it to you.
All right. Thank you.
Yeah, I'm not going to be able to stay up late tonight.
No,
I
I
was
mean,
up till
I'm
4
not. Neither,
o'clock
neither
this
am I.
morning going through
Ne-
my photos so that I can make
I'm
these
not
posts.
expecting you to produce this thing tonight, so no
Oh,
worries.
oh no, I'll find it for and email it to you tonight. I'm just saying, just on the side. I'm probably going to leave once we close out the taxonomy discussion. I'm probably
That's quite all
I'm
right.
going to go to sleep. So that was a good one, I thought. Oh, here's a rare species, Entoloma nodule. What is it? Nodulosporum, nodosporum, nodulus. It's a weird Entoloma that there used to be a genus Pausarella, and another one is Flavoviridae. And these are like these scaly, hairy little entelomas. They're not that common. None of these species are really that common. In fact, Flavoviridae, I believe, is in the Fundus Challenge, and I don't think this is much more common than that one. So the spores are very big and lots of angles. And as you might expect from the name, what is it, nodulosporum? I think the spores they're kind of irregular, bumpy. They have like nodes, little bumps on them. So they look almost like Inocybe spores. But this is there's no longer a genus Pausarella. There might be a Entoloma subgenus Pausarella. I'm not sure, but so I certainly sampled this.
That's cool. They're like entolomas with Inocybe caps, fibrils on
Yeah,
the
and the
on the
spores
cap.
even look a little like Inocybe.
Yeah. It's like that. It's like a weird cross between the two.
Yeah, it is. Now, maybe I should have smash mounted some gill just to make sure there's not like either great big Pleurocystidia or like palisade of Chylocystidia, but I don't think I need to do that. I know what this is. This is this Entoloma nodosporum. So I found it before. I have at least one other observation of this from, I don't know, like 10 years ago or something.
Yeah, that's fun. You find that thing and you're like, oh, it's an Inocybe. It's, looks like an Inocybe, I assume in situ. And then you put it on the scope and you're like, oh, I don't know. What
Yeah.
is this thing?
Well, actually, I didn't pick it. One of our club members picked it. We have a club member, her name is Grace, and she's mainly interested in finding edibles, and I have to really keep an eye on her because she wants to eat everything. So, but she goes out, she finds really interesting things. We also found, I think she may have found, or I may have found it, I forget, Psilocybe caerulipes. That was found on Sunday. But that's actually pretty common, so that was not a big deal. This is sort of a big deal, this one.
Yeah, and if you have to rein her in for eating that one, you can't let her out of your sight.
I don't think she would have eaten the. Now she always asks me, but she always asks her first question is always, can I eat it? But that's okay. A lot of the club members are like that. But she will go out and look for weird things too, that little things that she knows she's not going to eat. Just it's only a matter of time before her interest becomes elevated above just wanting to eat everything, because you can see that. I mean, she wouldn't have brought these things to me, if she was only interested in eating them. I mean, look, they're very small. They don't look very appetizing. So.
Cool.
Look at the size of this thing. He can hardly fit it on the computer screen. Look, there's a penny. So this is actually a Amanita section vaginate. It's one of these things in what Rotulus calls series penetratrices. And I think, I got some DNA sequences on some of these penetratrices things that I sampled a month or two ago. There's some names I didn't recognize. I'm going to have to look. I think maybe the Canadians have published names, other than the ones Rod was using. So his names are like penetratrix, penetrans, and pseudopenetrans. I'm going to have to do a little research on that and see if these are the same things that are now going by some of these new names. So of course I sampled this thing. Why would I not sample this thing? It's really interesting. So these guys have really long stalks that root in the ground. You have to dig them up. This one wasn't really all that deeply rooted, but you can see it's, the vulva's got dirt all over the soil all over it. So it was buried, but not as deeply as some of them. These things can be buried up to like 10 inches. So the soil was very soft where this one is. And this one's interesting too. It's pretty white. Look at that cap, is very white. See the little umbo in the middle of the top of the cap? That's one of the features associated with series penetratrices. Now there's other vaginate that have umbos, but this is like a really abrupt small umbo, and it's very hard. And it's like the battering ram that the mushroom uses to push up through rocks and around roots and stuff without, the cap getting damaged. So that was a pretty interesting one. Usually I find all of these penetratrices things in oak woods generally. So this is a trichoglossum. Now I was hoping to be able to put a species name on this based on the number of septa on the spores. But it looks like, I hope we don't have a mixed collection here. I don't think so. I didn't pick them. Oh, there's a pretty cool ascus. So I got a picture of a nice filled up ascus.
Oh wow, the spores are kind of laying on top of each other. Or?
Yeah, they're not arranged like in any kind of neat, one after another. There's a word for that, and I forget the word. And there's a word for this, where they're just kind of all jammed in there. But I forget the word. Oh. So the spores seem to have variable a number of septa. And I did a little research on trichoglossum, and some of the species do not always have the same number of septa on the spores. And I think there's one species where it's like 9 to 14 septa or something like that. Now, how do I know this is trichoglossum?
Are these from a spore drop, Dave? Because immature spores on trichoglossum.
This is from a drop.
Okay. Yeah.
Yeah, this is from a drop. But I also have a photo here somewhere, and this is through my other microscope, and I edited this. Now, this one that's second from the left, I think I can probably count the septa in that one pretty well. And so I edited this photo. I really tried to beef up the detail to be able to see the septa. But how do I know this is trichogloma, you might wonder? Well, there's another photo here somewhere that shows the setae. There we are. This is at 100 times. Those spore photos are at 400 times. So you see these little pointy things sticking out all over the place. This was not from a drop. This was just from a little bit of the surface on the upper stipe that I scraped off and smash mounted. And those are little hairs. So this is 100 times magnification. You can actually see with a loupe, you can see little like brushiness, on the surface. But it's better to make sure that you're seeing not just like some some velvety sort of covering that you're actually seeing bristles, it's a good idea to get a little bit of material under the scope and look at you only need 100 times magnification to get a really good look at. And the setae, those are called setae. There's other kinds of mushrooms that have setae. There's, I think there's like a marasmius that has setae on the gills. So, and they do look like those also.
And the spores are on the gills, like this.
Well, this is on the marasmius, the setae would be on the gills. Yeah.
Okay.
Yeah, I think it's marasmius coherens. I think it's coherens. I think that's the one with the setae.
Cool.
I have to review that.
And the setae indicate that it's trichoglossum.
Yeah, geoglossum doesn't have these little pointy things all over the surface.
Okay.
Yeah, they're like little bristles.
Cool.
And actually, I think geoglossum are a little bit easier to pinpoint the species because I think most, if not all of this, well, actually, I'm going to say something that's wrong here. It's in the same ballpark as what Igor was calling an error a little while ago, but not really based on DNA, just based on actually knowing that there's like a lot of different species in these geoglossum and trichoglossum genera. But if you use a field guide, which I suppose is a dubious sort of thing to do, or a species index like on Microquebec, with geoglossum, it is at least sometimes the case that a given species will have spores that always have the same number of septa. But the trichoglossum seem to have variable number of septa on the spores.
Well, it's so getting a spore drop from a trichoglossum, getting mature spores through the setae can be difficult. But
I just lay the thing down on a slide overnight and get up the next day and there's spores. I usually
but they're not always mature. Notice how we couldn't make out the actual septations, the septa, not setae.
That just might be the resolution on my microscope, but they're from a spore drop. I mean, the spores that I photographed are from a spore drop.
Yeah, but sometimes the mature ones get caught up in the actual setae. So it is like you said, it is more reliable to get spores from a spore drop from a geoglossum that doesn't have the hair. Just something I've noticed when working with these things.
Yeah, well, when I read descriptions of trichoglossum species that I found on Microquebec, the numbers of septa on the spores were, I think there was only one species where it was always the same, or where they say it's always the same. The other ones would give a range of number of septa. Like there was one that was like 9 to 14 or something like that.
And that 9 to 14, that would be probably a rare find because most of these things are. You should use the Malcolm Greaves key. It's much better, and I respect Quo tremendously for even trying to work through this. But it's much better than Quo's. There's 10 times the species on that website. So.
Yeah, I think I have that key. I think you actually gave it to me during one of these sessions.
Yeah, I found at our Thompson Town foray, I found Trichoglossum farlowii, which I think you found also maybe this year. And that thing, it looks like it looks difficult to discern, but once you find a mature spore and can really make out those uh, septa, um, it's very clear that they can be confusing when there are immature spores.
All right, I'll keep that in mind. Oh, Jason, are you still there? Okay, I put up something in the chat for that polypore looking thing that Jason posted a while back tonight, and it's actually Calipostia guttulata is the thing I was going to propose, but it's supposed to grow on conifer wood, so I don't know. It might be wrong, but this does look kind of like what he showed, right? It's got the potholes in it.
And that the texture of that thing, I assume you found this before. Not super dense,
Not
or
rock hard, but pretty dense. It's hard to pass a knife through it, but you can.
yeah.
So I think
Yeah.
Michael Quebec says it's supposed to grow on pine, but I found mine on hemlock. So it might grow on a variety of substrate.
It's funny. A lot of descriptions that I found on Micro Quebec list things that are found with hemlock, but we don't have hemlock here. So,
Right.
are they
So you find,
associating?
yep, you find it
Are
in
they?
other substrate. Yeah, I
Yeah,
know.
and is it just associating with a different conifer, or what's going on there?
Yeah, I really think it's you're onto something here that um the habitat for or preferred substrate for a given species seems to vary regionally with what's available in a given region, and but or it might be that there's more than one species, you know. So, but back to the discussion about it looks like we can use this name here, uh, cal was it Calcipostia gutulata.
Yeah, and what is the best name to apply, right? That's that's where I come back to. Uh, if there's a good species to apply, I think use it. But there's a case to be made for Igor, just put it at the genus level or
Yeah, the problem is, I like the idea of AFF period. That's actually, a way of 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 suggesting that there's some doubt, that's but the problem is that those types of names are not available on INAT. And I don't think they're available on MO either. At least on MO, you can propose